Today's bioprocess bench spaces consist of vast arrays of devices and technologies from an ever-growing list of providers. In general, these systems are impressive custom assemblies but fundamentally lack two important qualities: the reliability of an automated sampling system for process analytical technologies and a comprehensive software solution to integrate and execute all singular end-to-end workflows, ensuring full data integrity and traceability. Secure Cell Technologies have already answered the call to address these limitations by developing our ingenious process monitoring software, Lucullus, and its analytic hardware companion, Numera, a state-of-the-art automated sampling system. Both Lucullus and Numera were developed specifically with the other in mind and engineered to work as a seamless network-driven bioprocessing dream team. Implementing these innovations, Secure Cell has now achieved the next benchmark in our efficiency mission, and we are excited to introduce the next generation of laboratory productivity, the IntegraOne Modular BioLab System, the first and only fully automated solution for bioprocess development and small-scale production of therapeutic proteins, cells, and vaccines. IntegraOne covers all relevant process steps, upstream to downstream, and provides all necessary analytical tools to quantify metabolites, their products, and record their key attributes with the utmost confidence. IntegraOne is the synthesis of Secure Cell's technology platforms which have been brought to industrial maturity over the last 28 years. Utilizing our greatest minds and scientific breakthroughs, IntegraOne is a total solution for true end-to-end functionality. Let's meet the system. The precious cargo of each BioLab solution is the product, and IntegraOne reinvents how we cultivate, sample, analyze, and harvest biomass. The system's immaculately designed multifunctional reactor base, or MFR, contains each bioreactor and consolidates an unprecedented variety of functionalities in a single unit, including infrared heating, cooling with ambient air, stirring, weighing, UV lighting for algae cultivation, a sensor connection panel, and an integrated microphone and camera to record events. The Opera Module 1 acts as the MFR control station and aligns the functionalities of up to 4 bioreactors, governing parameters such as pH, dissolved oxygen, CO2, N2, antifoam, aeration, and volume, and also serves as a communication gateway for Ethernet, Modbus, and USB devices. The system's low-volume sampling capabilities are made possible by the Sapphira probe. Its silicon membrane, available with pore sizes from 0.2 to 32 micrometers, allows you to collect low-volume filtrate samples from 0.1 to 2 milliliters from each bioreactor, which are gravimetrically managed by the Opera Module 2's liquid flow control with an accuracy of plus or minus 0.01 grams. The samples are then routed to our patented Crunchy Sampler, where they are preserved in ice water and protected from evaporation. Each sample is then sealed and assigned QR codes autonomously. For continuous processing via perfusion, the filtrate is routed to temperature-controlled harvest bags, where its raw products are collected and await redirection to downstream processing. IntegraOne is designed for perfusion rates of 0.5 to 1 reactor volume per each 24-hour cycle. After collection, content from the harvest bags is periodically pumped to the amphora station for weighing, then sent to the Protein A downstream device for affinity chromatography, purification, and cool storage. Other methods will be supplied soon. Amphora scales are also used in unison with the pump configuration of the Opera Module 2, ensuring consumption of all feed solutions, base and antifoam is gravimetrically controlled and balanced. A variety of multi-feed options for each scale are available in order to support diverse processing needs and parameters. Subsequently, each reactor's feed flasks are automatically monitored and replenished from their corresponding media refill bags stored in the IntegraOne refrigeration units. All airflow, oxygen, nitrogen, and carbon dioxide are managed by the mass flow controllers in the Opera Module 3 and linked to the system's pH and dissolved oxygen sensors. Low-volume samples drawn by the Sapphira probe can also be routed to the MetaMaster, where glucose concentration is measured by a highly accurate and state-of-the-art photometric method. As a consequence, glucose concentration can be controlled at any level by pre-installed control algorithms. Numera draws samples from the bioreactors according to the programmed schedule and transfers them to the routing module. Drawn samples can then be diluted and filtered before they are passed on to the station's analytical device of choice, or to be stored in the autosampler for later analysis. Lastly, the Opera Module 0 is home to Lucullus, the software conductor for the IntegraOne orchestra. Lucullus initiates and visualizes all processing cues from planning and preparation through execution and evaluation. Its programming allows each lab to operate a smooth end-to-end process, streamlining each unit for groundbreaking efficiency. How efficient can IntegraOne be? Let's review what a 20-day rundown looks like for 8 reactors running in 24/7 operation mode with the IntegraOne 830 during a 20-day cycle. Every 2 hours, a sample is drawn, processed, and transferred to an analytical device, totaling 240 processed and analyzed samples per reactor, or 1,920 samples per system. Every hour, a sample is cold stored for further analysis, totaling 480 cold stored samples per reactor, or 3,840 per system. Glucose is also quantified every hour, for a total of 480 glucose measurements per reactor, or 3,840 per system. These 3 results alone reveal the potential continuous monitoring has to accurately control each stringent parameter of the system, effectively safeguarding all operations throughout the entirety of every bioprocess run. During such a run, the exacting nature of a single IntegraOne system saves a minimum of 48,000 manual actions. This number represents the repetitive tasks performed by highly skilled laboratory staff, amounting to the full-time equivalent of 15 to 18 technicians. Each IntegraOne installation eliminates this wasted physical labor and the costly mistakes that come with it. Furthermore, it multiplies the amount of insightful data collected while ensuring its traceability and integrity, specifically by minimizing human error. In comparison to traditional setups, IntegraOne provides the highest quality data generated at a fraction of today's labor effort. And thanks to the integration power of Lucullus, each IntegraOne system can be interconnected and supervised off-site, securely controlled from anywhere in the world in real time by authorized lab personnel. Every component of each system works in perfect unison. Elegant by design, its scalability features will revolutionize industrial lab environments like no system before it. The IntegraOne is a collective hive, unlocking the resources of time and space through efficiency, with a product line perfectly positioned to lead the future of decentralized bioprocessing and cell therapy.